Welcome, everyone. Thank you for joining us today. This is the first of two webinars on Dutch elm disease. The first one here is going to go over the biology and really how Dutch elm disease works. And we're very fortunate to have Tom Prosser with us here, the founder of Rainbow, and truly a pioneer in the industry, especially with Dutch elm disease. Today, it is worth one ISA certification, continuing education credit. If you did not submit your ISA number when you registered, please enter that into the question and answer or the chat, and we'll take care of that and get those submitted for you. Today, I will be supporting Tom with manning the chat and the question and answer. A quick request for everyone is to, if you have questions, please enter them at the Q&A, which is at the bottom of your panel. That way we can make sure I can check off, make sure that we answered all the questions and we don't miss any. During the session today, I'll be, if there's a question that's particularly pertinent, I'll interrupt Tom and ask the question. Otherwise, we'll take some of them at the end. But if there are questions that are just a quick clarification or a rate or something of that nature, I'm more than happy. I'll just type that into the question and answer to answer that right away. If there's any technical difficulties, please be patient. We'll get that squared away. If with any technology, if the internet drops off, just stay on the line and we'll get it back up and running as quickly as possible. And if there's any questions or you need any additional support, just please message me. And with that, I'll kick it over to you, Tom, to kind of go over what today's about and everything like that. Thank you.
Great. Thanks, Chris. Appreciate that. Yeah, like Chris said, ask questions. A lot of times, some of the best information that is given during this is from questions. There's just so much information that one could learn about this. I've been doing this. I've been working with Dutch elm disease. I worked with it for probably about 30 years. I haven't been really actively working with Dutch elm disease the last 10, other than working with like the city of Evanston and some of these and some clients. But I spent many years in the field trying many different things. And when we first started working with trees, it was Dutch elm disease, and a lot of failures in the beginning. We tried a bunch of different stuff that did not work until we were lucky enough to come across work done by a master's student at the University of Minnesota, as well as a professor from Canada. And basically what this program here today is, purpose is to give you that information so you don't have to try a lot of different things yourself, but also to give you like key distinctions and key information so that you start to have common sense about what to do, because Dutch elm disease is one of the more complicated diseases that you're going to deal with. And it's also one of those diseases that had many years of no success in terms of managing it. So we're going to show you what we've learned, and we'll go from there. So just to start out, I love to show this picture. This is a photo of trees in Minneapolis. That's a road called Newton Avenue. And this neighborhood signed up all their elm trees on their boulevard, got all the neighbors involved. And we've been treating these trees now for 40 years. And I think they lost one in a storm. But other than that, all the trees have lived. The philosophy and the methodology that we're currently using is very effective. And again, I'm going to show you that, like I said in this. Hey, Chris, I'm trying to get this control panel, oh, there we go, out of the way here. How do I do that? Anyways, like I said, this is our philosophy and how we run our program. So, oopsie, sorry about that. So another very large account that we took on back in the early 2000s was the city of Evanston. And the city of Evanston is known for their very large trees, and they were losing many trees a year. And they were losing actually about 600 to 800 trees every year. And I got called in and we started working with the city, and we were able to put a program together and implement it. And after we implemented it, they virtually lost no trees. In fact, I think they lost, it says 14 elms between 2004 and 2015 after losing 600 to 800 per year. And then in 2016, some new foresters came in and they stopped following the program, and right away the disease started to go up. So we've been in there again and we've got them, so they're not losing their trees. This is a great example of a very effective program. So first of all, I want to go through some of the basics, and I know a lot of you guys know a lot of this stuff, but I'm going to just start at the very beginning. What is Dutch elm disease? First of all, it's extremely contagious, and if it's not managed, it will literally wipe out all the elms in a population in 2 to 3 years. It's so contagious, and that has to do with how the beetle spreads the fungus, which is basically, the fungus is called an ascomycete, and ascomycete is a type of fungus that's very similar to like bread mold and so forth, but this particular fungus only grows in the vessels of elm trees. It's very specialized. Nature is pretty amazing in that way, how organisms adapt. The primary way that this disease is spread is through a beetle that will feed on a living elm tree after breeding in a dying elm. If that elm tree that the beetle emerged from had Dutch elm disease, the spores of the disease are all over its body, and basically this beetle's life cycle revolves around elm trees. So it'll go up and it'll feed, and I'm going to point out some important distinctions as we go, but it feeds in the 2 to 4 year old branches of the tree. So that's an important note to make, because that's how the disease gets into the tree at least 90% of the time, or most of the time. We'll talk about the other way that it spreads, called root grafts, in a little bit, but most disease is spread by this beetle that's feeding in the 2 to 4 year old branch crotches. And like I said, they breed in dead and dying elms, and most of these beetles flying around are not carrying the disease, because much of the dead elm or dead elm wood or dead branches are not infected. But if that tree did die from Dutch elm disease, all those beetles that are breeding in it will be carrying the fungus. And that's why it spreads so quickly, is because one elm tree can produce thousands and thousands of disease-carrying beetles that are flying off to feed on a healthy elm tree. So symptoms of this disease appears in the foliage, and actually the fungus is growing inside the tree in the xylem, and as it grows, the symptoms emerge or are shown. An important distinction to understand is that the symptoms do not always reflect the disease. And that's another thing to put as a note. You have to check the inside of the tree to see where the fungus is. And we'll talk a little bit more about that in a bit. There's different weather patterns that will cause the symptoms to look different year over year. So another key distinction you look at is, what do the leaves look like? There are a lot of things on an elm tree that create yellow leaves, but Dutch elm disease has a its own very unique look, in that you get curled and droopy leaves, and oftentimes they'll fall off the tree. Flat leaves in the tree are typically not Dutch elm disease. So you got to be looking for that curl. Typically you'll get some brown with some yellow and some light green, can all be mixed in. The disease itself, again, is the fungus, right? And it's growing inside the tree, and it grows in a very specific pattern. It's not like a blight that will cover it, but elm trees and all trees are very compartmentalized, the xylem are. And that's, a self-protection. It's a defense mechanism in the tree to have highly rigid compartmentalized cell walls. Well, that forces the disease to grow straight down. And typically, as you can see in this photo, the disease will go straight down the tree in a very narrow band. if you look at the picture of the tree on the right, where you can see that band is, we basically removed the wood, and you can see how narrow it really is. Once it grows down and hits the roots, it then spreads out and goes systemic in the tree, and it'll come back up the tree. Like the photo on the left, you can see the original disease coming down was maybe an inch or two wide, and then it hit the roots and it came back up, and it was like 6 inches. And it also, the tree root grafted to itself, so the fungus is growing back up the tree. Typically, what we have found is once it hits the roots, we cannot ever save the tree. However, if we can catch the fungus before it hits the roots, we have very good success at saving these trees. But I will say, injection alone does not save trees, and we're going to cover that. Even though you might see labels and people put out information that you can save trees that are infected, that is a myth. Like I said, I've been doing this for many years. I've tried dozens of different things, and unless you isolate the fungus from the tree, you're not going to be able to save that tree. And we're going to talk about that. It's a process called tracing. It's the only way we've ever saved trees. And again, I say that because a lot of people try and save trees with injection, and we'll be talking about injection, but it just makes the symptoms go away. Another key distinction that you need to understand about elm trees, and not all trees are built like elm trees. All trees are built differently. But in the case of an elm tree, the only xylem that is functioning in the tree transporting water is the current year, which grows in the spring. So last year's xylem the tree plugs it up and walls it off. So you can tell, when the tree got the disease by looking at where the fungus is. And you can see in this particular log where the arrow is, where the disease came down the tree in maybe last year's xylem tissue. And you can see that it's coming back up. It's gone systemic in many places. Another thing to look for is there's a very, very characteristic reaction the tree has to Dutch elm disease. Now, the brown coloration is not the fungus itself. It's actually the tree's reaction to the fungus. Again, that's another important distinction to recognize in creating a common sense approach to this. You have to realize that the brown colorations, the piloses, and that becomes more important when you're trying to save a tree because, again, you're trying to, in saving elm trees that have this disease, you have to be able to isolate all the fungus. So recognizing this is key. We'll talk about this more later. So I don't know if there are cities on this. I don't know if there's colleges or golf courses, but people who have large populations of elm trees. This is typically the program that we recommend is these really 5 steps. We have a very large population of elm trees that we manage, and so we treat that like in the Twin Cities, very similar to this. So these are the 5 practices you need to understand in order to save the trees. This is a great picture, isn't it? You can see how old those cars are, and then it's the same street. So the first is scouting. You have to know what's going on, and you have to periodically look at your elm trees, your valuable elm trees. This is what cities do. Cities don't typically treat all their trees, and so they're out scouting because you need to catch the disease before the beetles breed and hatch and spread it massively. So if you've got a large population of elms that you're protecting or working with, and you're not treating them, we recommend that you monthly view them at a minimum during the growing season. The other thing is, we work with a lot of golf courses, and they might have 100 to 200 trees on their property that we might treat over a 3-year period. We will go in and look at those trees on a monthly basis. And the other thing we will do is we will go outside of the golf course a half mile to a mile and see if we can find diseased elms, etc., and report them to the city or homeowners to try and get them taken down. The third reason that you do the scouting is for trees that you've treated. and we do it in the third year of protection. We're going to talk more about that. We're going to talk about how the protection works and why that is important, but I won't cover that at this time. So removal of the disposal and dying elms is very important for spread, as we've said. And I call this a tale of two cities. Now, again, these pictures are fairly old, and I've been giving this presentation for quite a few years, 15 years or so. But back when there was a big fight for Dutch elm disease, there was a big difference between the way the city of Minneapolis fought it and St. Paul. St. Paul ignored it. And as a result, they lost about 99.5% of their elm trees very, very quickly. City of Minneapolis, on the other hand, had great sanitation practices. They scouted their trees. They found it, they got them out very quickly. In fact, so quickly that in like this particular case, I came and looked at these trees, I could have saved them, but they were so fast to remove them that we couldn't. But again, that's why the city of Minneapolis has 30–40% of its elms left. Even after 50 years of fighting this disease, they still have quite a few elms left in their population. Again, thousands of disease-carrying beetles can emerge from one elm tree. So this sanitation is very critical in terms of making sure that you don't have a lot of pressure of beetles around you. And if you're treating trees, again, it's important. We'll talk about why, because you don't want 1,000 beetles feeding on your protected tree. Even though it'll probably work, there are little gaps in the treatment we're going to talk about, so that you'll have an understanding of what the treatment does, how it works, how long it works, what you need to be careful of, etc. So this is a key. The other way that Dutch elm disease spreads is through what's called root grafts. Now, root grafts form between trees of the same species. So you're not going to get an elm tree grafting to an oak tree. You only have elm trees grafting to elms. But more interestingly is, if elms are significantly genetically different, they also do not graft together. But what happens is the fungus will grow down the tree and into the roots and up the next tree and kill it very quickly. So you have to take root grafts into account and manage root grafts if you have trees that you're working with, especially if one's got the disease. Or if you're protecting a population of elms and there's some elms that you're not protecting, but you either need to remove those trees so you don't get the root grafts, or remove them, or put the root graft in. But again, here's another key distinction that you have to recognize, and that we'll say again: if the fungus comes in through a root graft, there is no protection that you can inject into a tree that will work. At least we haven't found anything. And Dutch elm is a very different disease than oak wilt, so you can't compare the two. But it doesn't, if you get root grafts and you have a disease, you have a tree you've been protecting, and there's another one that's not protected, you got to take that into account, because if that gets it and it's root grafted and it goes into the tree, your treated tree will still die. Lots of ways to sever root grafts. I'm not going to cover that. Some of these techniques are covered in other programs, but we use trenchers, we use air spades, especially if there's utilities, we'll use an air spade to cut the roots. Other distinctions: roads disrupt root grafts. I've never seen a neighborhood road that had root grafts underneath it, where the disease passed from the elm across underneath the road. So you already have that there. However, if you're dealing with sidewalks, they do not disrupt root grafts, so you have to take them into consideration. Now, I'm going to show you a slide here of a plan. So you can see the diseased elm tree, and what we would do is check the tree to make sure, first of all, can we save the tree? And we trace the disease, and we would see where it's coming down. So as you know, as we just learned, the disease is coming down the tree in one inch. It's going to be on one side or the other. And if you can catch that disease, and we'll talk about tracing in a minute, you can save that tree. However, if you trace it and it's in the roots, you have to put in root graft barriers. And this is how we do it. Again, I'm not going to go through all of it, but you have to get all the roots. And one way to reduce root grafts is to grind the stump. Cut the tree down, grind the stump. But one warning is, if you have the disease in the roots and the disease is growing towards another elm tree, you have to cut those roots before you cut the elm tree down. Otherwise, those disease spores will get sucked into the other tree very quickly. Only in that direction. And then if you don't cut the stump, if you don't grind the stump out, it will grow around the stump, right? So you either have to get that rid of that stump or put root grafts in on the other sides of the trees to protect the other trees. Again, not going to go in detail of it. You're not going to remember it anyways, but just know you have to deal with root grafts. That's a very important part of it. All right, let's talk about saving disease elms. So there are 5 key distinctions that if you understand them, it makes it easy. This is not rocket science. It's just understanding how the disease grows, etc. So before we talk about it though, a lot of people, and there's people that are selling things out there that make a claim that they can save disease elms with injections. I know right now it's kind of surprising actually. Propiconazole is still out there and being touted as a curative, even a preventative. We're going to talk about that. And we'll also talk about the work we did in the 90s on propiconazole with Syngenta. Anyways, but I just want you to know that I have tried every combination of injections many times, trying all different things, injecting every year, injecting multiple chemicals, injecting, etc. And it doesn't work. And there's a reason it doesn't work. Is as you saw that column of disease coming down the tree, that xylem is blocked. And so you're not going to be able to get funded to the fungus. So it literally will grow down into the roots. However, what will happen is it will appear that you've saved the tree because the symptoms go away. The leaves fall off, there's no more symptoms. And that's just because you're blocking the fungus from continuing to grow. But it is growing. It's growing microscopically in the tree. And what'll happen is 2 to 3 years later, the whole tree will die. And it'll come out in the spring with a full set of leaves, and then you'll see it just it looks like it just fades out. It looks a lot different than a normal tree dying from Dutch elm disease. So the only way that, and we've saved lots of trees this way, the only way is through this tracing method. So the first thing again is the fungus only grows in the current year xylem. That's the key, because that means you can isolate the disease. You don't have to carve it out or anything. You just have to isolate it on the sides. The other one is the brown discoloration is the tree's reaction to the fungus. And the fungus is often growing ahead of the tree's reaction. So we typically say 10 feet max. And so when you're tracing a tree, you get to the end of the stain, it'll tail out. You go another 10 feet down the same xylem tubes, which you can figure out by sticking a nail into the xylem. It'll literally light up the xylem in both directions just by putting a sharp point into it. So you can follow the same pattern down the tree that you're cutting. And this is what it looks like. So you're cutting down this tree and you're cutting along the fungus. Now, this is a great photo for showing the fungus, but this is one of those trees that it didn't work on. And you can see the fungus grew right into the roots. We were just late. We've saved hundreds and hundreds of elms with this process. However, not every year is a great year to save these. Typically, if you have a dry spring, the fungus grows slow. And the symptoms in the tree are very much closer to the actual growth rate of the fungus. However, if you have a very wet spring, what'll happen is the fungus grows really fast and you hardly see any symptoms. I was doing a demo of this process to the Illinois Arborists Association back a number of years ago. And we were gonna, we found a tree that had literally like 2 feet of wilting leaves in it. And so we were gonna show this tracing process, and we did, we showed it, and that fungus was all the way in the roots. Well, it was a wet year that year. So, you know, there are years that we'll go out and we'll have 30, 40, 50 trees that look like minor symptoms, and we're not able to save any of them. Then there are other years where it's dry, and we'll go out and start saving those. We'll go out and start to save these trees, and we'll be able to save virtually all of them. So again, the weather is a big factor, unfortunately, in this. So again, you know, the disease grows down the tree in a narrow band, which is what you're trying to isolate from the rest of the tree. And once it grows into the roots, it cannot be saved. We've never saved one. Yeah, like I said, I've worked with this many years. I tried saving some really glorious trees. And we have saved some really glorious trees. This is courtesy of Alex Shigo, this diagram here. And what this diagram is showing you is how the vascular tissue is connected to branches. And at least in elms, but in most trees, the vascular tissue is only connected on the bottom. So stuff coming up. So you don't have vascular tissue on the top. And that way you can always find the disease in the tree by going to the base of the branch that is diseased and starting your trace there. And it'll always grow straight down. Now, if you have a codominant stem, sometimes that has got more vascular tissue connections on the sides. So you have to watch out for codominant stems. A codominant stem is 2 branches that are the exact same size. They're not really branches, they're both stems that are codominant, meaning they're the same size. So the trunk might go up and then it breaks into 2 equal sized branches. It's pretty rare to have the fungus grow down the middle. It's almost always on the outside. So this is kind of a little bit of review. So you don't need a deep cut into the tree. You can see in this case, there's the branch, it's on the outside, and we were able to, the fungus grew down you know, little ways, we were able to finish it. Now, I want to warn you on something. See those cross cuts? That's operator error. That's injuring the tree more than you need to. You only need cuts along the side, not down the middle. But again, it's something that you still need to cut across because you need to pound the bark off. Usually you just take a hammer, hit the bark after you've cut it, and it pops off. So like you can see on this tree, you can see how close the fungus is to the ground. So this is actually a stage photo, I'll admit. But what we're doing is we're cutting a trench here because there's a tree next to it. And it's just good practice. Here's some examples. Again, there's that same tree. Here's one, you can see the fungus, see the burl up there? And look at the fungus, it came down into the burl and actually shot back up pretty wide. And then it popped out the other side. We were able to save this tree for quite a while. You can see even they re-landscaped it. But you know, it really does work. Here's that other tree. You can see it's starting to heal or seal itself up and wall off the old wound. Now, some trees are not good candidates to be traced. Trees that are in really poor condition and are not gonna, it's just not worth it. So you can see poor recovery on that tree that we traced versus good recovery. And then this is our first tree we ever traced in 1989. You can see the massive size of this tree behind the house. In today's world, this would probably be a $5,000, $6,000, $7,000 removal. So, you know, you can tell people were very, very happy what we did. Also, we overdid it here. We went a little wider than we needed to, and you can see the cross cuts in there again, unnecessary. So let's hop over and talk a little bit about protecting elms. We've tried many products, and we did a pretty major research project I'm going to talk to you about, but we only use thiabendazole, which is, the brand name is Arbotect. It's made by Syngenta. And when you're treating trees, there's one purpose and really one purpose only. And everything you're doing with this treatment is about this, and that is to evenly and completely cover the crown of the tree with enough fungicide so that all the 2 to 4 year old branches are covered in case a beetle comes to feed that's carrying the fungus. If you do that, this protection is virtually 100% effective and works extremely well. We've got, like I said, thousands of elm trees we've been treating for many, many years. And, we have golf courses where we have 200 trees on them. And in 40 years, we've lost 1 tree or 2 trees total. Not many. You do lose some. But typically, if you do, it means they didn't get even complete coverage. Or in the 3rd year, we're going to talk about the 3rd year in a minute. But let me keep moving on here. So back in the 90s, we did a very sizable study. We had a major highway that was going to go into renovation, and in the cul-de-sacs or the turns, where people would get on that little freeway, there was probably about 100 elm trees in there that the Department of Transportation told us they were going to be removing in 10 years. They had that planned out that long, and they gave us the trees to experiment on. And what we did is we treated the trees with at the time there was 4 different products that were making the claim: Arbotect, or thiabendazole, propiconazole, or Alamo, or I don't know. There's other products out there now. Propojet or something is one of them that's out there. Another one was Fungisol, which is basically 2 milliliters per injection site, and then a product called Phyton, which was making claims. And what we did is we treated the trees, and then we went in, and for 3 years in a row, we injected the fungus into 30 different places if the fungus would grow. And what we found was, first of all, Fungisol, nothing. Every site grew. Didn't work. Way too little. I mean, when you're putting Arbotect in the tree and it's a 20-inch diameter tree, I believe you're putting in about 30 gallons of solution. So they're putting in, like an ounce in the whole tree. Phyton, it's very difficult to get in the tree, and it didn't. I don't know if it didn't move, it didn't work. Again, there was no protection with Phyton. Propiconazole, however, or Alamo, Propazole, or whatever it is called, for 1 year after treatment, it worked. It was 100% of the 30 different spots that we did, it worked. And same with thiabendazole. The first year it worked 100%. However, in the 2nd year, the propiconazole, it was like it was not even in the tree. We had no protection. Again, the injection sites where we put the fungus all grew. However, the thiabendazole, again, was 100%. We got no growth. So Arbotect, we knew lasted 2 years by this time. We knew propiconazole only lasted 1. We knew the other products didn't work at all. So then we go to our 3rd year, and we did it again with the Arbotect trees only at this point. And what we found was 15% of the places that we put the fungus, the fungus grew. 85% of the spots it didn't. But what that told us was the Arbotect or the thiabendazole was starting to wear out in the tree, and you didn't have enough in certain places. We thought, well, maybe we should put in more, and we tried that. It didn't work. And in future studies. So basically, it got 85% crown coverage in the 3rd season after treatment. This is why we scout all of our trees in July of the 3rd year after treatment. So I'm going to talk about that in a minute, about that scouting, but there's some keys. I want to just cover some key distinctions about treating trees, especially, and I believe this is true for any injection that you do. You know, if you go into the trunk, you know how the fungus grows in a very narrow, rigid line? Well, that's how your product is going to go up the tree, in a very narrow, rigid line. You're not going to get very good distribution. However, there is a researcher in Canada, his name's Earl Swayze, and he discovered that, and this was back in the 60s, so quite a while ago, that if you put materials into the tree, into the root flares, that you get very wide coverage. Apparently, when the tree brings in moisture and it shares across the tree in the root flares, and where the roots come into the flares and shares. So all treatments should be in the root flares to get the best, even and complete coverage of the tree. And that's important for a few reasons, actually. Like I said, the trunk tissue is hard and rigid. You don't get good distribution. However, if you go into the root flares, you've got more surface area, so you can get more injection sites in, and you do less damage overall to the tree by the treatments. Again, root flares shares, but also meristem, which is, cambium is a meristem, the subapical meristem on the tips of the trees is meristem, where you have cells that divide. Trees, they don't heal, they seal, right? They grow new tissue, and that's created by wherever the meristem is. Well, there's more meristem in the root flares, there's a little more, in fact. And so the holes seal up much quicker. I look again at microinjection. I know it's a little bit unbelievable to me, and they put things on the market. But there's, any microinjection that you would use that claims that they get even and complete distribution of a fungicide is basically, untrue, to put it kindly. You got to use macroinfusion. Now, macroinfusion is a large amount of solution into the root flares. So, like a microinjection might put in, maybe a quart of material into the whole tree, whereas macroinfusion and a large tree might put in 20, 30, 40 gallons. So again, the whole purpose is even and complete coverage of the 2 to 4 year old branches. Hang on a sec here. I wanted to make sure people saw the research. I haven't had this one in my slide deck for a while, but I understand that propiconazole is, people think because propiconazole works on oak wilt, it works on Dutch elm. Oak wilt's a different animal. Elms are different animals. I mean, oaks are different animals than elms. Oak wilt's a very different animal than Dutch elm. And even though we get good protection with propiconazole for oak wilt, even root grafts, doesn't work in elms more than 1 year. And there's a problem with that, in that, there's 2 major cycles to a beetle that spreads this. One is in the spring, and that is before full leaf. And when you're treating and you're injecting, because the xylem, like we talked about earlier, only transports water in current year, you need to have full leaves on the tree to treat. And so you've already had beetles feeding in the tree and the fungus going in the tree, and as we know, that fungus gets going, you come in and treat, it's not going to work. The symptoms are going to go away. So, if you're treating something every year with something that works for one season, again, you're not getting protection in the beginning of that year, and you very well could be hiding disease that you could trace out by just going in and treating. So we don't recommend this. There was a big, in the 90s, there was a big effort to say that this fungicide worked longer. And we found out the hard way, but so did a number of companies. But, use our lesson to not make the same mistake yourself. There is a study, and if you're looking for it, we can send out the link, but it's Virginia Tech, Blackburn. This is the link to the paper. A master's student went in and literally tested for propiconazole in the tree, and they found that it only lasts for 7 months. And that's pretty much backed up what we were finding in the field. All right, so let's talk a little bit about why, people say, well, why don't you treat every 2 years? Well, it's the wounding. Whenever you're injecting a tree, you have two kinds of wounds. One wound is the drill, and the drill literally removes tissue from the tree, which of course is a wound, and the tree has to respond to that. But then when you put any chemical in, you get what's called, I'm trying to think, maybe someone can help me with the right word, but it's like a necrosis, right? Around there where you've got high acid or high chemical, and it literally kills the tissue around the injection site. One of the reasons why we use a half gallon of water per ounce of Arbortect is to not have that necrosis, and that works. If you're putting anything that's, doesn't, have some water with it, you're almost likely to get necrosis depending on the, the features or the, of whatever that chemical is, if it's highly acid. In the case of Arbortect's highly acid. So, that's why we put it in the water. So, but, the other thing is, if you inject trees too often, you get a buildup of wounds, and they'll continue to build. And at some point, the tree will start to fail. What we found is that if you treat the trees once every 3 years with an injection, at least an elm tree, the wounds are buried deep enough in the tree that it's like you're putting wounds in for the first time if you do it every 3 years. That's most trees. And so we elect to, instead of treating all our trees every 2 years, which is expensive, we elect to go a third year and then go scout the trees. And sure enough, we go look at all the trees. There'll be about 20 trees per year. Maybe 2,000 now. I'm not sure what our exact numbers are, but and we scout them in July, mid to late July, and we look for the fungus, look for disease, and if we see it, we'll go and trace it out quickly. And then that's a tree that doesn't have the disease, right? And so that way we're being able to avoid over-treating. Now, there are certain situations where you have a lot of disease pressure and you have a lot of disease-carrying beetles. In these cases, if you treat every 2.5 years, and I would limit that. I wouldn't do that forever, but if you've got unmanaged elms around you and there's a lot of beetles flying, this is not a bad thing to do, which means you treat like in June, August, early September. And that way you're protecting, you've got that tree protected at least in the following spring too, right? So that's a way, that's a practice that I think is fine. So 2 times in 5 years, right? So there's that. Now I want to talk a little, one more slide on macroinfusion. There are pitfalls, right? And the pitfalls have to do with not getting the material where you want it. And it has to do with making sure and these are that. So I know there's going to be another program coming that Chris Haugen is going to be presenting on how to inject. And basically, I would make a card like this so that your crews have it. If you have slow uptake on a tree, typically the average elm tree, no matter how much material you're putting in, 40 gallons, 50 gallons, 30 gallons, 20 gallons, 90%, 95% of those trees should take this up in less than an hour. If it's less than that, odds are you don't, you either the tees are pounded past the current year xylem, you have clogged tees, you might have unequal pressure. If you have poor water quality, you're in trouble. The water, your water will be white, and it'll, the material crystallizes basically. So anyways, you want a razor sharp drill bit because you want to cut open the tissue to let the material in. The only other situation where it seems to be slower is if you have a very dry, droughty year and the xylem tubes are smaller. So with that, I'm done. I see there's a lot of questions possibly.
Yeah,
Chris,
thank you, Tom. Yeah, we got a few questions. So one of them came over the course of the last 40 years. have you seen an upward or downward trend in the rate of infection from Dutch elm disease in the Twin Cities?
Well, it's an interesting question because where I live right now is the western suburbs. And I remember in the early 80s, Dutch elm disease out here, and there was no elms. Literally every elm had it, and there was none. And I moved out here about, 15 years later, and there's small elms. Now you got big elms around here, and it's like coming sweeping through again. I had 3 beautiful elm trees. I thought maybe they're immune. Well, they weren't. They all died about 3 years ago. There's just not as many elms. So, the disease is definitely around. I mean, and you can see it when you drive around right now. You can see the wilting, browning leaves on the trees almost for sure. At this time of year, it's going to be Dutch elm. But if it's an elm, it's for sure Dutch elm. So, I don't know if it's gone up and down. It's, back in the day when there's a lot of elms and there's a lot of beetles, you had a massive number of trees dying. So, but yeah, you definitely have diseased elms around and disease carrying beetles. So one way you could look at this is the number, they used to trap beetles and then test what % of them are carrying the disease. And I would guess that the % of the beetles carrying the disease these days is significantly smaller. But then again, there's a significantly smaller number of elm trees to feed on for these beetles. So, okay, does that answer your question?
I believe it does, yes. Another question was just a quick one. How many trees were in that Dutch elm disease challenge study along the highway in Minneapolis? Do you recall?
Yes, each treatment had I believe 20 trees. I don't know, there was like 150 elms in the whole thing. I think we took 20 of each. And I think we highly suspected the fungicide wasn't going to work. And I think we limited that to 10, if I'm right. We didn't want to go with 20. But with the Arbortect, propiconazole was the big new thing back then. And there was a lot of claims like this was going to replace Arbortect. It's easier to treat. It's faster. And we were excited, like, wow, we could have something faster and easier. But, so, I mean, that was those, it was really between those two. I don't think we believed the other ones actually worked, and we were right, they didn't, but,
There's a question around trenching. It was, how much of a trench do you need to have? Do you trench around the entire tree if it's suspected of being infected and you have adjacent trees? Kind of,
yeah.
Is it just to separate the two?
You got to use a little geometry. So, you got to make sure, if your elm that's diseased is surrounded by elm trees, yes, you got to go around the whole tree. But if there's an elm tree over to the side, you kind of got to look at it like, each tree's putting out a fan of roots. And you basically want to, you go around, usually you'll go like a semicircle around the diseased tree, halfway around. So it's pretty unlikely that you're going to get root grafting. Well, that pretty much covers it. So you don't have to, you don't have to do the whole tree in all cases, and in most cases even, just in between a semicircle, like a half circle.
There's a couple questions shifting a little bit to cultural practices. With all the cultivars of elm claiming to be resistant to Dutch elm disease, do you think it is a safe practice for municipalities to consider these cultivars in their planting inventories? Or will these trees also require an eventual scouting treatment plan?
So here's what I understand, and this is, again, I don't have personal experience with this. It's just my research. When they were creating these immune elms, basically what they did is they found trees that should have gotten the disease and didn't. And they took, clones of those, and they started breeding them with others. And then they would challenge them in the nursery. And the most they could ever get with any of these so-called immune trees or resistant trees was 90%. So 10%, they could get it up to 90%. So all these trees that are out there, 10% of them are susceptible. Now, there's a kind of tree called the Liberty elm that was promoted back in the day by this company that, I don't know how reputable they were. I understand those trees are not, that it's much lower, a much lower percentage of those trees are immune. So I know that if you can identify the Liberty species or Liberty elm species, which are now fairly sizable trees, I know that our practice at Rainbow is to treat those. But when you're talking about like cathedral elms, and I'm trying to think of a bunch of the other names, frontier elms, and 90% of those. So I always tell people, if you're going to plant them, plant 3 of them. Then you'll for sure have 2. So does that answer that question thoroughly enough?
Yeah, and there were a couple of them around there. I think you mentioned one individual was asking your favorite cultivars of the resistant varieties, and I think you mentioned one of them.
I'm not the right person. Maybe there's someone else on this that might want to chime in on that.
Okay.
I haven't seen enough of them. I know our consulting arborists at Rainbow deal with a lot of them, and someone like Jeff Rick is someone who would probably have a favorite, and he'd base it on, does it get leaf minor? How's the branch structure? How's the, how's the form, right? Does it break? How well is it growing? And I don't know the answer to that, so sorry.
No, absolutely. A question here kind of around the scouting is, do you monitor monthly and trace if the disease is present every year or just the third year? Can you maybe just clarify around the monitoring program and then also what you trace?
So we do a monitoring program where there are a population of unprotected elms. Amongst a population of protected elms, we do monthly, and that's golf courses, etc. Right? Like the city of Evanston, they send someone out and they're scouting because they don't treat every single tree. They treat like a couple thousand, I guess. But protected trees are the ones that you look at every third year, and because we know they're only 85% coverage in those, and that there might be some possibility of a beetle landing on the wrong spot. So does that answer that? Like back in the day when I was a consulting arborist, I literally once a month I had 9 golf courses, and I had the state capital, and I would take 2 days or 3 days, and they'd pay me, and I would scout them, and then I'd drive the neighborhood around to look for diseased elms. My first scout would always be, it'd be in people's log piles. Just trying to reduce the pressure. So that was a long time ago, but so that's my answer. I hope that answers that, right?
Absolutely. And following that up with, there's a question on the tracing procedure, and can you maybe just describe the goal of that tracing program is to reduce the spread of the actual pathogen within the vascular tissue. Can you just describe how you're judging that outcome?
Yeah, well, it's the fungus grows in a linear line down the tree, right? Maybe an inch wide, and the purpose of it is to find it, and then to follow it. You cut the bark off, in a narrow band down the tree until you come to the end of it. And the goal is to prevent it from getting into the roots. So, you find it, and then, if let's say you're 20 feet above the ground and it tails off to the end, well, then you go another 10 feet down that same xylem column, and you make a cross cut at the bottom so that it can't get any further. And then you go cut in 1 inch all the way along the side of that. And you're basically making that tree disease free. You pull the bark off, expose it to the elements, and that fungus dies. So it's like cutting out cancer, right? You cut it out, it can't spread. So if you miss any of it, the tree will still die. So you can't miss it. So, the purpose is to get it all.
Fantastic. Shifting a little bit, there's a couple questions on other Dutch elm disease, more fungicide or additional products that are out there on the market. A question from Guy here is, has there been any use of Dutch trig in the United States? Any research to support it?
No. I think the best example of Dutch trig was the city of Denver. Denver was using Arbitect for a lot of years, and Dutch trig came in, and I talked to the forester. This is a while though. Dutch trig is, I don't even know if it's around anymore, but Dutch trig was, they used it on 500 trees, and the city forester told me that the trees all got kind of sickly from it, but also he said there was no difference between those 500 trees and the other trees. Basically what Dutch trig is, it's a pathogen. Is it a weakened Phytophthora pathogen, I believe? But they,
verticillium.
Is it? Okay, yeah, it's one of those diseases that it's like it grows in broccoli or something, and it doesn't really grow in elms, but it's still that pathogen, and they inject it into the tree, and supposedly the tree's defensive response is so big that if Dutch elm disease were to come, it would, the tree would protect itself. So it's a way of stimulating the tree to protect itself, and I just don't think it worked. I mean, it didn't work. Again, we would have loved it if it did, because it takes about 5 minutes to treat every tree. So you could treat every tree every year. But again, we don't believe it worked.
Thank you.
Yeah. By the way, there's a professor at the University of Minnesota. His name was David French. He's no longer, he passed a number of years ago. He told me one day, he said, there's been over 500 purported cures for Dutch elm disease. And he said the only one they've ever seen have effectiveness is thiabendazole. And thiabendazole is fairly unique in that it can move into new wood and it's very long lasting. So it's got a half-life of like 450 days. And so that's why it lasts in the tree, and the fact that it moves into new wood. So, I mean, you wouldn't believe the things they used to try back in the day. I mean, used to be that, 80%, 90% of all the street trees in the whole northern United States was elm trees. So, I mean, to have lost all those, I mean, they tried a lot of things. And, most things just don't work. It's a very aggressive fungus.
Quick question here for the first time treating. I assume you treat as soon as you have good uptake from full leaf to first signs of fall coloration.
Yes.
The second time, do you retreat the same month, or do you try to move it up over two or three treatments to ultimately treat all trees in the spring? So maybe just talk a little bit about kind of timing treatments.
Yeah, you can treat the trees anytime from full leaf to fall, and that's fine. A key, again, part of the treatment is you have to look in the tree very thoroughly to make sure there's no fungus growing, because it literally will disappear after you treat, and it'll show back up 2 years later. So, it doesn't matter. If you treat it in June of, let's say, 2018, you can treat in June of 2021, and June of 2024 is fine.
Perfect. And a quick plug just for myself, this coming Thursday, at the same time, I'll be doing a webinar on the macroinfusion process, and we'll go in depth. Tom showed those kind of the quick pitfalls. We will go through those step by step to ensure that you're not making any of those mistakes, and you're really getting to the nitty gritty of those little details so that you get all of your applications done in a timely manner, and really you're getting that predictability of the protocol.
Right. Yep.
Are there any additional questions from here? I really thank you all for all the wonderful engagement that you've had here and all the acknowledgments as well. It makes these webinars much more enjoyable. So if that, thank you again, everyone, for attending. I have your ISA certification numbers. We'll get those entered in to get your credits here. Sometimes they do take a few weeks for ISA to post them, but we will get those posted, and then you'll have at the end of the day or tomorrow morning an email from us with the recording, the link to the recording. So if you want to watch it again, if there's something you missed, please do. And certainly you have Tom and my emails as well. Please don't hesitate to reach out to us. We want to support you and help you out as best we can wherever you are. with that, thank you, Tom. Have a wonderful day, everyone, and be safe, and have a wonderful week.
Great. Thanks, guys. Thank you. Bye.
Thank you.